Officer at A very good evening and a warm welcome to all the doctors present here. I am Devolina from Clinic. We are very much proud to be a part of this webinar as a digital partner. Clinic is India's largest live digital CME and doctors-generated medical content platform, which is exclusively free for all the medical practitioners. So now with all of you, I am going to share a short glimpse of Clinic. Thank you so much all for your patience. You can always find us on Google, which is www.clinic.com. There we have live sessions conducted by eminent speakers across the globe. You can read our MedWikis, which is medical Wikipedia for doctors. Besides, you can use our telemed site, and in your leisure time, you can play polls and quiz also on our plan. So now, without further delay, I would like to hand over the session to Dr. Hamida Mustafa ma'am to start with our today's program. Over to you.
Thank you. Good evening and a very warm welcome to all. I'm Hamida Mustafa, doing my pre-final year MBBS in Sirvadur Medical College and state coordinator of SNO TAMILNADU. I will really elated to see students and doctors from across the length and breadth of this nation have joined this webinar. IMA-JDM has been doing a plethora of webinars on various topics with objectives of disseminating knowledge and keeping the medical community informed and updated. IMA-JDM, in collaboration with SNO Tamilnadu, under the guidance of Dr. Abul Hassan Sir, presents the webinar on the topic, How Do Your Life Begin? A journey through the human embryonic period. The patrons of our webinar are Dr. Abul Hassan Sir, a well-acclaimed pediatric surgeon from Europe. Sir is the chairperson of IMA-JDM Tamil Nadu and also the IMA-TM State President-elect 2024. I would organize this. For today's session, a community family medicine PG from AIMS partner, also General Secretary of IMA-JDM Tamil Nadu. I feel honored to introduce the speaker for today's session, Dr. Stephen Francis, MD DNB Anatomy and postdoctoral fellowship in cytogenetics. He did all his courses in CMC Vellore. He served in the Department of Anatomy at CMC Vellore for 8 long years. Then he served as an AP at Madras Medical College and Vellore Medical College. Now he is a senior assistant professor at Government OD Medical College, the Nilgiris. If you have any queries, kindly leave it in the chat box below. All the queries will be addressed at the end of the session. So without wasting much time, I request Dr. Stephen Frank, sir, to take over the session. Thank you, sir.
Good evening, everyone, and welcome once again. Thank you for those kind words, Hamida. And my session is the embryonic period. The embryonic period actually is from 3rd week to the 6th week, to the 8th week of life. Actually, some books will call it up to the 10th week of life, 5th to 10th week. That is when almost all the embryonic processes, well, almost all the organs and the organ systems in our body are completely formed. But before we step into the embryonic period, I want to go into the early 3 weeks of life, and that would be my presentation for the next 1 hour. And hopefully I will do justice to this, because the way God has created us is too amazing and too fascinating for us to capture in 1 hour. All right, let me share my screen. All right, can you see? I hope you can see. We'll start with the first week of development. We start today's class with 2 small cells, the oocyte and the sperm cell. The oocyte at the time of ovulation is in the first meiotic division. It's just completed and it's in that stage. And the secondary oocyte is about to undergo the second meiotic division. At this stage, the oocyte has entered the fallopian tube and the fertilization occurs in the ampulla. If the ovum is not fertilized by this time, the uterus absorbs or reabsorbs this ovum. Just by this, we can understand that the oocyte actually has a very minimal time period where it is available for fertilization. The other small cell that we have today is the cell called the spermatozoa, or the sperm, and it's from the seminiferous tubules. And after spermiogenesis, They're stored in the epididymis where they get matured, and through this tract, which we all know, they get deposited in the female genital tract. At the time of deposition, there are around 600 million spermatozoa in the vagina, but only 1% of them enter the cervix. And it takes about 2 to 7 hours for these sperms to move up to the fallopian tube, up to the ampulla. This movement is caused by its own propulsion and the uterine cilia that propel it towards the ampulla of the site that is ovulated. Spermatozoa, by the time they reach, they become less motile by the time they reach the fallopian tube. And they cannot fertilize the ovum until they undergo two important steps called the capacitation and the acrosome reaction. Now, capacitation is, I like to joke about it saying, it's like removing the coat of the sperm and ready for fertilization. And it's because of the conditioning of the female genital tract, and this capacitation period lasts for 7 hours. The coat that is removed is the glycoprotein coat, and the seminal protein coats are removed from the surface of the acrosome. Only capacitated sperms can actually penetrate the corona. So if there are sperms that are incapable of capacitation, that sperm or the entire semen deposit gets wasted. So the viability of the gametes, the oocytes are viable inside the uterus or inside the fallopian tube for 12 hours, whereas outside they are viable for 24 hours. Whereas the sperms are viable for 48 hours inside the female genital tract. Fertilization, probably the most important step, and probably that's the thing that has gotten me excited over many years, is when the sperm regains its motility at the time of ovulation because of the chemical attractants released by the corona radiata. And this causes the sperm and the oocyte to actually start coming together. And then at the end of fertilization, there's intermingling of maternal and paternal chromosomes. This is a scanning electron microscope picture of an ovum with many sperms that are sticking onto it, and that's how it actually is. Many sperms actually reach the site of fertilization. Only one fertilizes the oocyte. And the stages of fertilization are divided into 3: penetration of corona, penetration of zona, and the fusion of the oocyte and the sperm cell membranes. Penetration of the corona radiata occurs because of 3 things, mainly hyaluronidase, tubal mucosal enzymes, and the movement of the tail. Capacitated sperms, because of the lack of glycoprotein, can pass freely through the corona cells. Then there's a second step is the penetration of the zona pellucida. The zona is a glycoprotein shell around the oocyte. And this, it facilitates the sperm binding and induces the next reaction called the acrosome reaction. Capacitation was one, and now we have acrosome reaction. This is mediated by the ligand ZP3. And the acrosomal enzymes are acrosin, neuraminases, and esterases. When the head of the sperm is in contact with the oocyte, the cortical granules that are inside the oocyte get released into the plasma membrane of the oocyte, preventing further sperms to penetrate. That is why, even though there are so many sperms at the site of fertilization, only one sperm can enter the oocyte and cause fertilization. So that's how it happens. The sperm plasma membrane remains behind, and the head and tail enter the oocyte. At this stage, the oocyte actually undergoes some very interesting, important changes because the completion of second meiotic division occurs. This was actually held and arrested in the metaphase stage, and now only the second polar body is extruded. And this nucleus is now called the female pronucleus. Only after the sperm enters does an oocyte undergo its second meiotic division. Until then, the oocyte is suspended in the meiosis 1 metaphase. So now we have the male pronucleus and the female pronucleus, and on day 1 of your life, the zygote is formed. The female pronucleus brings with itself 22X and the male 22X or Y. And at the time of fertilization, the following 3 things are established: restoration of diploid number of chromosomes, determination of chromosomal sex, and initiation of cleavage of the zygote. For 24 hours, the zygote or the pronuclei are in contact with each other and the nuclear material mixes. I think that's a very important step. We can take a moment to pause and understand what happens in the first 24 hours of your life. That's when the two pronuclei chromosomes mix together, and then there is an establishment of a new entity, which we know and we commonly call as zygote, but that new entity is a new human being there. The typical mitotic division occurs now on day 2. That means all these cells, that means the male and female pronuclei, are gametes, which are only tuned to undergo meiosis. Now, suddenly, this zygote has now changed its entire chromosomal division and the cell division format into a mitotic division format. And in the day 2, the first mitotic division occurs. And the cells keep on dividing until you have a 16-cell stage called the morula. The morula is from a term called the mulberry appearance, because it appears like a mulberry. The zona pellucida is still there. On day 3, that is third day after fertilization, these cells become smaller and each division is called blastomeres. I did not mention a thing in the previous slide, that the first division, mitotic division occurs, is not entirely equational. There is a slight mismatch of the chromosome, of the cytoplasmic entity between these cells. Anyway, now we have blastomeres. The blastomeres is essentially a stage before day 4 where the blastocyst is formed. In day 4, these blastomeres start organizing themselves into cells that are going to form the future placenta on the periphery, and the cells that are going to form the future baby or the fetus or the embryo in the center of the structure. So this is how the embryoblast will be inside of the morula, and the trophoblast will be in the outside of the morula. Now, at the end of day 4, at the term end of day 4, fluid from the uterine cavity, by this time the embryo has entered the uterine cavity, and the fluid from the uterine cavity now passes inside the morula and creates a cavity inside the morula, and that cavity is called a it's called a cyst, and this whole structure is called the blastocyst. This is called the blastocyst structure, where we have a cavity here called the blastocele, and the cells aggregated in one area called the embryonic pole, and the opposite area will be called the abembryonic pole. And you can see that the trophoblast cells now take up a flattened form. The trophoblast cells take up a flattened form. Yeah, so that was embryonic pole or the animal pole and the abembryonic pole. Now, these trophoblastic cells or trophoblast cells have this property to stick. So if the zona pellucida isn't there, the trophoblast will stick wherever the zona pellucida has left it. If it has left it in the uterine tube, and that would be resulting in an ectopic pregnancy. These trophoblast cells also have a property of burrowing or eating up or burying into. So if you let the trophoblast cells stick in the anywhere else other than the uterine cavity or inside the uterine cavity as well, they'll keep burrowing through the epithelial structures there and even go through the wall of the structures. That is the property of trophoblasts. Now this golden structure called the zona pellucida, I'm calling it golden because it's quite important now. It starts to disappear by day 6. That's the one that prevents this trophoblast from sticking actually. So by day 6, the blastocyst, day 5, the zona pellucida starts disappearing, and by day 6, the implantation starts after fertilization. So that is how we have an implanted embryo inside the uterine cavity. And the most common site of uterine implantation or embryonic implantation is in the upper part of the fundus, superior part of the posterior wall near the fundus. If it implants anywhere else, we have an ectopic pregnancy. Well, this is again a clinical part of this first series of slides. where we can have a prevention of fertilization at various levels. We have contraceptive methods at the vaginal level, at the uterine cavity level, hormonal level at the ovary, even permanent ones at the fallopian tube. If fertilization is not possible in the normal way, then there are assisted reproductive techniques where the sperm male pronucleus is injected directly inside the female. You can see this injection happening here. That is just a suction apparatus to hold the ovum in place. This is the polar body, actually. We know this. I just skipped this part. We'll go to the summary of events of week 1. Day 1 was fertilization. Day 2 was 2-cell stage ending in morula. Day 4 is the blastocyst, and the embryo enters the uterine cavity. Day 5, the zona pellucida starts disappearing, and day 6, implantation occurs. By the end of your week 1 of life, the embryo is already implanted. We'll just skip these questions. Yes. The second week of development is actually called the week of 2s. By the end of the first week, we have this structure, right? We have an inner cell mass and an outer cell mass and the blastocele. By day 8, what happens is the trophoblast differentiates into 2 layers and the embryoblast differentiates into 2 layers. That's why we call this the week of 2s. Now the blastocyst is already embedded into the endometrial stroma. These are the 2 layers of the trophoblast. We have an inner cytotrophoblast and an outer syncytiotrophoblast. The cytotrophoblast is the inner layer. It's mononucleated. There are cell margins that are clear, and there are mitotic figures that are present. Whereas the syncytiotrophoblast is a multinucleated zone with indistinct cell margins, and there are no mitotic figures. You can see in this picture as well where the embryo is embedding inside, and you can see the syncytiotrophoblast just burrowing through, and the cytotrophoblast is in this layer. The inner cell mass, this is the outer cell mass was trophoblast. Now the inner cell mass called the embryoblast also differentiates into 2 layers. We have this pink zone here and this fusiform zone here. We call that fusiform zone as the hypoblast layer. That is a layer of cuboidal cells. And the outer layer or the pink zone here, we call it as epiblast, and they are columnar cells. So we have now a bilaminar germ disc. We have trophoblasts in 2 layers that are called syncytiotrophoblasts on the outside, cytotrophoblast on the inside. And on the inner cell mass or the embryoblast, we have 2 layers, the epiblasts that are columnar on the embryonic pole. And towards the abembryonic pole, we have cuboidal cells called the hypoblast. Now, there are 2 cavities that are going to appear. We already had only 1 cavity called the blastocele now. So in the epiblast cells, there is a cavity called the amniotic cavity. You can see that the amniotic cavity is a very tiny cavity in this picture. This is the roof of the amniotic cavity called by cells called the amnioblasts, and the floor of the amniotic cavity by these pink cells called the epiblast cells. That's the amniotic cavity. And you can see in this picture, that's the amniotic cavity. So these cells would be the amnioblasts, and these cells here would be the epiblasts. These are columnar cells. These actually don't have or don't retain the columnar shape. They get slightly flattened, maybe low columnar to cuboidal, and that's the amniotic cavity. This is the other cavity, which was the initial blastocele. Let's see what happens to that. What is this one? This is the syncytiotrophoblast. You can make out that there are no cell margins in the multinucleated zone. This here is the cytotrophoblast. That here is the epiblast. That's the hypoblast, amniotic cavity, and we still have the blastocele hanging there. So this is the day 8 embryo. We have a bilaminar germ disc, an amniotic cavity, and a blastocele still present. But day 9, the blastocele is more deeply embedded, and if you were to scope the uterus at this time, you would see a fibrin clot at the place where the embryo has embedded inside. These spaces that you see in the syncytiotrophoblast at this time, there are gaps that occur. Those are called lacunae. So such a day 9 embryo is called the lacunar stage embryo. Now let's see what happens in the blastocele, or what does this hypoblast do? I like to comically call the hypoblast cells being a very needy and very copycatting kind of hypoblast cells. Now these cells say, okay, amniotic epiblast has its own cavity. I want to have my own cavity. And these cells of the hypoblast migrate along the cytotrophoblast here. This dark bold line here, that's a cytotrophoblast. This outer zone here, that's a syncytiotrophblast. And the hypoblast cells migrate on the inside of the cytotrophoblast and form a new cavity. That cavity is called the primitive yolk sac. And these cells are what contribute to what we know as the Husser's membrane or the exocoelomic membrane. This here is the Husser's membrane or the exocoelomic membrane, and the green pointed arrow is actually called the primitive yolk sac, and that replaces the blastocele. Now we have this bright red zone here as the exocoelomic membrane or the Husser's membrane. This here, the cytotrophoblast. That there is the syncytiotrophblast with the lacunar stage. And then you have the amniotic cavity surrounded by the roof of amnioblast and the floor of epiblast, and the primitive yolk sac cavity surrounded by the hypoblast and the Husser's membrane. That is the end of day 9. This is the same picture as the previous one. All right, you can go ahead and find out what those are. Days 10 through 12, the blastocyst gets completely embedded. And the surface epithelium defect is like totally closed up by the surrounding germinal epithelium of the endometrium. And there's a slight bulge that you can see on the uterine cavity. And by day 12, the uteroplacental circulation is established. You remember this picture where you had lacunar stages? These were actually the maternal sinusoids, because the maternal uterine arteries divide and keep dividing into what we call as the uterine sinusoids. And these are filled with maternal blood. And that maternal blood opens up as this keeps burrowing inside. The maternal blood capillaries or the sinusoids of the mother open into the blood opens into the lacuna of the fetus. And that is the establishment of the uteroplacental circulation. This obviously occurs more at the embryonic pole, as you can see in the picture as well. At the abembryonic pole, what happens? There's not much of syncytium actually, because the burrowing is happening on the other pole. There is not much of syncytiotrophoblast on the other side. Well, so now we have the maternal sinusoids that are dilated capillaries, and we have the syncytiotrophoblast embedded, eroding into the maternal sinusoids, and the lacunae become continuous sinusoid, and the uteroplacental circulation is established. This is day 12 of your life. In day 12 of your life, that's when the embryo stops depending on its own internal fluid of the blastocele and starts depending on the maternal sinusoidal blood for its nutrition. All right, now we come to an interesting component called the extra-embryonic coelom. My pointer right now is playing in the extra-embryonic coelom. We have here, let's start from the outside, maternal sinusoids, maternal endometrium, syncytiotrophoblasts and the sinusoids opening into the lacunae. Cytotrophoblast is light green cell-like outline layer. Then we have this thick layer that is called the extra-embryonic mesoderm. This is the first time a derm layer has been mentioned. Mesoderm is a derm layer that allows for establishment of structures that are derived usually from the mesoderm. They include mainly the blood vessels in this case. This is a new layer that has formed from the cells of the yolk sac. And they go all around. They go even around the embryonic pole. They go all around. And that is the extra-embryonic mesoderm. So if you were to say what lies on the inside, the amnion and the Husser's membrane lie on the inside. And what lies on the outside is the cytotrophoblast. Those are the two boundaries of this extra-embryonic mesoderm. And it lies outside this embryonic disc. This is called the embryonic disc, by the way. The epiblast and hypoblast together is called the embryonic disc. This extra-embryonic mesoderm does not give rise to any part of the embryo. That's why it's called extra-embryonic mesoderm. All right. And now in this mesoderm, we see a cavity developing that's called the extra-embryonic coelom. Extra-embryonic coelom. Now this extra-embryonic coelom divides the extra-embryonic mesoderm into two layers. You can see a bright red layer internally and a bright red layer externally. This bright red layer internally is called splanchnopleuric layer, and the bright red layer externally is called the visceral, parietal or the somatopleuric layer. So that's how we have this extraembryonic mesoderm and the cavity or the coelom that divides this extraembryonic mesoderm into two layers, the inner visceral layer or the splanchnopleuric layer, and the outer parietal layer or the somatopleuric layer. The full name would be somatopleuric layer of extraembryonic mesoderm. The full layer of this would be splanchnopleuric layer of extraembryonic mesoderm. Well, if you can see, the coelom does not extend here, does not extend to this part of the that is cranial to the amniotic cavity. Let's call it cranial for now. We know it's not the cranial caudal axis, not established, but for simplicity's sake, for understanding's sake, the coelom does not extend into this space, and that would form the future connecting stalk. That would form the future connecting stalk. At the same time, the mother's endometrium undergoes something called the decidual reaction. That's called the endometrium of pregnancy, where the cells become large, polyhedral, full of glycogen and lipids, and there's intercellular spaces are filled with extravasate. The tissue becomes so edematous, and it's very easy for burrowing, very easy for burrowing. So at the end of day 12, we have uteroplacental circulation that is established. Extraembryonic mesoderm is formed, divided by the extraembryonic coelom, and the formation or the decidual reaction occurring. Day 13 of your life, we are still in the second week. The surface defect is completely healed. There may be a slight bleeding because as the embryo or as the syncytial trophoblast erodes into the maternal sinusoid, there may be slight bleeding, and that may be sometimes confused for wrong dates. And in the trophoblast, in the placenta, primary villi are formed. What are primary villi, you say? This is the structure of a primary villi. A villi where a villi is basically a tree. Okay, basically it means a tree or a projection. So you can see that there's this projection of what? Projection of cytotrophoblast into the syncytial trophoblast. That means the primary villi in its cut section would have a core of cytotrophoblast surrounded by a shell of syncytial trophoblast. That is how a primary villi would look like. All right. All right. Bilaminar germ disc in its development is much slower compared to the trophoblast development. We'll come back to that in a while. Before the end of the second week, there is a refinement that occurs in the cavity of the yolk sac, and that we call now as the definitive or the secondary yolk sac. So you see already we have the amniotic cavity, and now this big cavity was this primitive yolk sac. The hypoblast cells, they say, let's form a new, smaller, compact yolk sac, just like the amniotic cavity. So the hypoblast cells once again migrate and form a smaller cavity, and that is called the definitive yolk sac. What happens to the primitive yolk sac then? There can be fragments of that left behind, and we call that as exocoelomic cysts. We call them as exocoelomic cysts. Where is the extraembryonic mesoderm now? There, that gray line there, that is the parietal layer. And the gray line here, that is the visceral layer. And you see here where the coelom is not dividing the two layers, that would form the connecting stalk. That would form the connecting stalk. Yeah, so these are exocoelomic cysts, the pinched off bits of the original. This was the original blaster. This was the original primitive yolk sac. And a new hyperblast layer came like this and formed a new secondary yolk sac and remnants called the exocoelomic cysts. Yeah. That would give rise to the chorionic cavity. The secondary yolk sac or the definitive yolk sac is here. The amniotic cavity is there, surrounded by the amnioblast above and the epiblast below. And this cavity, which had the exocoelomic cysts, is now called the chorionic cavity. Yeah? Can you relate these two pictures now? Let's go ahead. And you remember the part where the extraembryonic mesoderm is not divided by the coelom? That would remain as the connecting stalk. What is chorionic plate now? Chorionic plate is nothing but the cytotrophoblast and the extraembryonic coelom coming together, getting stuck so nicely. That will be called a chorionic plate, or now what we know as a chorion. That is a chorion. That is that thing that is marked there, the arrow, that is a chorion. This lighter green represents the parietal layer of extraembryonic mesoderm. The darker green represents the cytotrophoblast layer of the trophoblast of the placenta. And this is the zone of, yes, syncytial trophoblast. So end of day 13, primary villi are formed. Secondary yolk sac is formed. Chorionic cavity and chorionic plate are established, and the connecting stalk is formed. In one day, all this is formed. In day 14, now the bilaminar germ disc decides to do something. This is the bilaminar germ disc, the columnar epiblast cells and the cuboidal or the cubicle shaped hypoblast cells. So that's the appearance of the bilaminar germ disc. There's nothing very great about it. But there, for some reason, a few cells in the hypoblast decides to become columnar, and that is called the procaudal plate. The procaudal plate. Now, the procaudal plate establishes what we now know as the head end of the embryo. The embryo was like this. There is no differentiation in the hypoblast. Suddenly, when the procaudal plate is defined, then that would be the head end. If the procaudal plate was somewhere here, that would be the head end and that would be the tail end. So the central axis is determined on the 14th day of your life. Your future tail end and your head ends are distinguished. So this is the procaudal plate. That was, what is procaudal plate? Columnar cells of the hypoblast established a cranial end. That's called the procaudal plate. But now there's something else happening in the epiblast cells. The epiblast starts to migrate its cells to the caudal end. The procaudal plate is because of the hypoblast becoming columnar. Now the epiblast becomes active and starts migrating cells to the caudal end of the bilaminar germ disc, and that we now know as the primitive streak. So the early embryo on the 14th day of your life had a procaudal plate, that is columnar cells in the hypoblast, and a primitive streak, that is columnar cells, amoeboid columnar cells in the epiblast or in the amniotic cavity. That is primitive streak. And the whole embryo starts to attain an oval shape rather than this circular shape that is drawn schematically here. So at the end of 2 weeks, we have a trophoblast differentiating into 2 layers, an embryoblast forming in 2 layers. The extraembryonic mesoderm forms and splits into 2 layers and 2 cavities in relation to the germ disc. That is why it's called the week of 2s. Some questions for the week of 2s. I'll just skip. These are the questions. These are easy to understand. I'm sure you know this by now. All right. Now in the 3rd week of life, that's my last section of the class today, I'm going to talk about changes that occur in the embryoblast separately and the changes that occur in the trophoblast separately. Embryoblast, we now know that till now, there's a bilaminar germ disc, a primitive streak, and a procaudal plate have been formed. The trophoblast, we now know that the primary villi is formed and the uteroplacental circulation was established. So what else happens in the embryoblast in 3rd week? The process called gastrulation occurs. Then there's the formation of notochord, the beginning of neurulation, the formation of the allantoic diverticulum. We'll go through each of these in detail. Gastrulation. This is where we left the 2nd week embryo. I've changed the shape to a slightly oval pattern because that's what it is. Procaudal plate was columnar cells in the hypoblast that established the head end. The tail end, the epiblast cells migrated and formed a thickening called the primitive streak. That is a 2nd week embryo. Now, what happens is by day 15 or 16, a groove develops in this primitive streak. This thickening of cells, epiblast cells all migrating to form this primitive streak. The buildup is so much that it starts to fall down, something like that. So a groove is seen. That groove is called the primitive groove. The nomenclature is just almost the same and it keeps changing, okay? So the day 15 and 16, 2 days, you can see the establishment of the primitive groove as well as something called the primitive node. So this I'm going to show now in this picture. This is the primitive streak. So what was that? That was the procaudal plate, and that depression there is would be the future buccopharyngeal membrane. The depression on the opposite side would be the future cloacal membrane. We're not talking about these membranes now. We're talking only about this thickening. Actually, I don't know if you realize, but you're sitting right now in the amniotic cavity looking down. This is the amniotic cavity. That's the definitive yolk sac cavity. And this is in the cavity of the amniotic space, or in the cavity of the epiblast, we see the primitive streak, this thickening, and this groove, that's called the primitive groove. The primitive streak actually is defined cranially by a bulge called the primitive node. Primitive node is also called the primitive knot, also called the Henson's node. And inside this primitive node, primitive knot, you can see, sorry, let me go back. You can see a depression, and that depression is called the primitive pit, or also called the blastopore. So if you tell me now what are the components of primitive streak, I would say primitive pit or blastopore, primitive node or Henson's node, and primitive groove. These three contribute to the primitive streak. Now, gastrulation first step is further migration of cells, of epiblast cells. So migration of epiblast cells will not stop till the end of gastrulation. It started at the ending of week 2, and will continue on till the end of gastrulation. So the epiblast cells continue to migrate towards the primitive streak, like so. And they achieve this amoeboid shape, become more flattened, have pseudopodia, and they bury beneath the epiblast. You can see that these hexagonal shaped cells are between the columnar cells and the cuboidal cells. So they're burying beneath it. All right, so you can see another picture to show the same thing. This is the epiblast cells, and the thickening of epiblast cells calls the primitive groove, and that's the primitive pit, and that's the primitive node. And you can see how more and more cells are migrating through and separating the two layers of epiblast and hypoblast. So what is the purpose of the groove? Let me go back a couple of pictures. What is the purpose of this groove? The purpose of this groove is to facilitate the falling off of the epiblast cells. So epiblast cells come to the primitive streak, they fall into the groove, and they push down the hypoblast cells and migrate down. So what is the fate of these migrating cells? Some displace the hypoblast cells, and they form a new layer called the endoderm. The others, so this is the endoderm now. This is the endoderm. Where is the hypoblast cells, you ask? They are pushed. Those cells actually lose connection with the support mechanism of the basal membrane of the epiblast, and they fall into the secondary yolk sac. Then some more cells migrate and take in the place between the epiblast above and the endoderm below. I'm not calling it hypoblast anymore because hypoblast does not exist. And those cells that migrate between the epiblast above and the endoderm below are called the mesoderm cells. And the mesoderm is now established. And the remaining cells that are of the epiblast are now termed as the ectoderm. So now we have 3 layers. All three derm layers are derivatives of the epiblast. And that's fascinating to me. Epiblast cells are columnar cells that migrate, burrow, displace, and form the 3 derm layers: the ectoderm, endoderm, and mesoderm. All right. This is a similar picture to show the same fate of invaginating cells. Such a trilaminar germ disc is now called the gastrula. A trilaminar germ disc is now called the gastrula. Gastrula was a term, I think, coined by a German scientist by the name Ernst Haeckel, I think, if I'm not right, if I'm not wrong. And he coined the term because he says it looked like a hollow viscous, like a stomach, with having when he cut it open, he saw the cavity of the amniotic cavity and the chorionic and the yolk sac cavity, and said it had 2 ends like a stomach, and he called it a stomach-like structure called the gastrula. The cells don't stop migrating here. The cells keep on migrating, keep pushing the or filling up the space between the endoderm and the ectoderm and establishing a fantastic layer called the mesoderm. This mesoderm is now called the intraembryonic mesoderm. But there are two places where the ectoderm and endoderm are not separated by the invaginating mesoderm cells. And those two places are the procaudal plate, which is the future buccopharyngeal membrane, and the cloacal membrane that is caudal to the primitive streak. So you have those buccopharyngeal membrane and the cloacal membrane are places where the ectoderm and endoderm are in contact. There is no intervening mesoderm. There is no intervening mesoderm. All right. Now we know that looking back at the embryo, we know that the buccopharyngeal membrane would later on form the oral cavity, and the breakdown of the cloacal membrane will form the anal opening and the genital urinary openings. You can see now that the mesodermal cells are not discouraged by the blocking of the buccopharyngeal membrane. They migrate cranial to the buccopharyngeal membrane. Can you see that? These cells are migrating cranial to the buccopharyngeal membrane. And this has a special importance. These mesodermal cells that migrate cranial to the buccopharyngeal membrane are called septum transversum. Septum transversum. All right. So that is how the mesodermal cells migrate. They don't stop there. They migrate into the connecting stalk. Now, connecting stalk was part of extraembryonic mesoderm. Now, these mesodermal cells are part of intraembryonic mesoderm. And you can see that in this picture from the very ancient Indrabir Singh, you can see how these intraembryonic mesodermal cells are actually migrating into the extraembryonic mesodermal cells. Essentially saying that the blood vessels that the extraembryonic mesoderm would develop as part of placenta, those blood vessels will be connected to the embryo through blood vessels in the connecting stalk. And we know that that would form the later on the umbilical cord. All right, now we come to the second part of the third week. You remember we left this in this stage. We had the primitive streak, a primitive groove, a primitive pit, a primitive node, and we had migrating cells that push down and form the three layers called the gas, and then that we know as the gastrula, or the process called gastrulation. And you know the mesodermal cells are migrated everywhere. But the epiblast cells, or these migratory cells, no longer called epiblast, I'm sorry, they continue to migrate into the primitive streak, but this time they won't migrate into the primitive groove. They'll migrate into the primitive pit. You can see that. If this was the epiblast cells and the hypoblast cells, and the migration would have started, at the end, you have this primitive pit, right? At the end of the primitive streak, you have this primitive node and the primitive pit. And you can see that cranially, the notochordal process is formed. Notochordal process is formed. That means cells migrate and form the ectoderm and endoderm. The cells also form a solid cord of cells called the notochordal process. That there is the notochordal, the green stuff there is notochordal process. This thing, this. And there is this breakdown of cells in the notochordal process and formation of a canal called the notochordal canal. You can see this. This is the procaudal plate, the cuboidal cells becoming columnar in the hypoblast, remember? And this is the primitive streak and primitive node. The primitive node has a primitive pit. And cells migrate through the primitive groove here and form the three germ layers. Simultaneously, cells migrate into the primitive node and go all the way up to the procaudal plate. This solid cord cells, or a solid cord of cells, that is called the notochordal process. And in this notochordal process, a groove continues inside, this pit continues inside, and that's called the notochordal canal. Now, this picture is what I want you to understand. We have the primitive node, we have the primitive streak and primitive groove here, the buccopharyngeal cloacal membranes here. And this process that goes cranially towards the procaudal plate or towards the buccopharyngeal membrane, that would result in the formation of the notochordal process and the notochordal canal. What happens to the notochordal canal? If you see the same picture, can you make out? It goes cranial. This is the notochordal process, this purple, and the cavity inside it called the notochordal canal. All right. So now we have this. We have, if you can imagine the cavity above, that would be the amniotic cavity, the cavity below, that would be the primitive yolk sac cavity. And there is no connection between the amniotic cavity or the yolk sac cavity. But suddenly the cells on the floor of the notochordal canal decide to disappear. And there's an establishment of a communication between the amniotic cavity above and the yolk sac cavity below through this hole or this opening called the primitive pit or the blastopore. And such a canal is called the neurenteric canal. Neurenteric canal is nothing to do with the adult gut, but because this is now probably the endoderm, that's why it's called neurenteric canal. If I were to draw a different cross section, epiblast cells, hyperblast cells. Well, now ectoderm, mesoderm, endoderm, notochordal process, notochordal canal, disappearance of the floor of the notochordal canal, and it's like this. This is the neurenteric canal. But that breakdown results in the formation of a new notochordal plate. You can make out this arch-shaped notochordal canal becomes fused with the endoderm, and it's called a notochordal plate. And from the floor arises a new notochord, and that's called the definitive notochord. That's called the definitive notochord. So now you see at the end of gastrulation, simultaneously notochordal formation, you have ectoderm, intraembryonic mesoderm, endoderm, and a definitive notochord in the center. Definitive notochord in the center. What happens to the notochord? Well, the notochord occupies the central position, and as the embryo enlarges, it lies in the midline, and it will give rise to two components alone in the adult or in the mature human being. That would be the nucleus pulposus and the apical ligament of the dens. The nucleus pulposus and the apical ligament of the dens. You remember this migrating cells going above the procaudal plate, that would be the region of the septum transversum. That's procaudal plate, that's developing notochord, there's the primitive streak, there's the cloacal membrane. All right. So mesoderm would not be there in the procaudal plate, would not be there in the cloacal membrane, and would also not be there in the region where just this place where the notochord is sitting. Just this place where the notochord is sitting. Here you can see that this is the intraembryonic mesoderm, and this friend here is the extraembryonic mesoderm. So at the edges of the trilaminar germ disc, the intraembryonic mesoderm is continuous with the extraembryonic mesoderm. Let's go to the third stage, in third process that happens in the third week. That's the neurulation. Neurulation is nothing but formation of neural tube, again derived from the ectodermal cells. And that is because of the stimulation of the notochord. This is the notochord. The notochord stimulates the ectoderm above to thicken to form what we know as the neuroectoderm or the neural plate. This is called the neural plate. And it thickens so much that it starts to fall down by its own weight. And that would be called the neural fold. Neural fold. This is the neural fold. That is the neural plate. This whole thing is the plate. And this bend here is the neural groove. Neural groove. When we do chick embryo studies, we can actually see this neural tube formation one day into the next. It's wonderful to see it. The neural fold continues to fall down by its weight, and the folds come together, and the epidermis is, or the ectoderm, sorry, ectoderm, I want to say ectoderm. Ectoderm is now separate from the underlying neural tube. So if you ask me, where does neural tube come from? I would say ectoderm. What part of ectoderm? I would call that neuroectoderm. Neuroectoderm is stimulated because of the formation of the notochord. Notochord stimulates the overlying ectoderm to form neuroectoderm, neural plate. The neural plate thickens, falls down as the neural groove, and there's a neural folds that come together in the midline to form or separate or pinch off the neural tube. There's an interesting cell here called the neural crest cells. They're nothing but these fold, this fold. These fold have specialized cells called neural crest cells. And when the neurulation is complete, these neural crest cells migrate into the entire embryo. The fourth step that happens in the third week is the formation of the allantoic diverticulum, actually also known as allantoenteric diverticulum. So this was amniotic cavity. This is the trilaminar germ disc now. The red line showing ectoderm, that yellow showing mesoderm, and the blue line showing endoderm. And this is the definitive yolk sac or the secondary yolk sac. A small outpouching of the yolk sac, a diverticulum, so to speak, enters into the connecting stalk. And that is called the allantoic diverticulum. All right. Now the embryonic disc itself starts to grow. And this keeps on growing even though the primitive streak starts to build up and then regress and shrinks and disappears. The embryonic disc as such keeps on growing. All right. So this is the growth of the embryonic disc into an oval or the slipper-shaped structure. Okay. And this primitive streak cells are, migratory cells for the epiblast. They migrate into the entire mesoderm. And there is a important, well, you can look up into other, now I don't have time, but we can actually discuss this on some other time, of all the factors and the molecular genetics that occurs in this embryo at this stage. There's establishment of anteropod posterior axis, there's establishment of dorsoventral, there's establishment of left and right, all because of these genes that are taking part, these proteins. A few of them are mentioned here. I'll skip this. I'll come back to the genes on my third lecture. Okay, just I'll remind myself to do it. All right, right and left axis. We'll come back to all this. The trophoblast, embryoblast had these four changes. We had a gastrulation, neurulation, notochord formation, and allantoid diverticulum. But the trophoblast had at the end of the second week or the beginning of third week had primary villi. That is the core of cytotrophoblast within surrounding syncytial trophoblast. That is cytotrophoblast. That is surrounding syncytial trophoblast. Then secondary villi is basically this extra embryonic mesoderm goes into and forms a new core. So the core is now formed by the extra embryonic mesoderm surrounded by cytotrophoblast, surrounded by syncytial trophoblast. That is establishment of secondary villi. Now by the end of third week, there is tertiary villi developed. So that is blood vessels inside the mesoderm. So primary villi on day 13 was cyto inside syncytio. Secondary villi on day 16 was mesoderm inside the cyto, inside the syncytio. Tertiary villi on day 21 is vessels inside the mesoderm, surrounded by cyto, surrounded by syncytio. What mesoderm is this, by the way? Extra embryonic mesoderm. Extra embryonic mesoderm. We take a section through like this. Primary villi on day 13 will look like this: center, cyto, peripheral, syncytio. If you take secondary villi, it will have mesoderm, and tertiary villi will have blood vessels, fetal blood vessels inside it. All right. I don't know. I thought I had a slide on it, but here first you can see if the blood between them. So this space is occupied by the maternal blood, right? And this is by the fetal blood. So the feto-placental or maternal blood and placental blood barrier is formed by syncytio, cyto, extra-embryonic mesoderm, and the endothelium of the fetal blood vessels. That would, four structures would form the maternal fetal barrier or the placental barrier. Syncytial trophoblast, cytotrophoblast, extra-embryonic mesoderm, and the endothelium of the blood vessels of the fetal capillaries. But as the tertiary villi mature, you see what's happening here? The cytotrophoblast cells start to disappear to decrease the gap between, or to decrease the placental barrier. And now we have the placental barrier only formed by two, the syncytial trophoblast and the fetal endothelium, fetal endothelium. So the placental barrier shifts as the placenta matures, So this is the same picture, but you can see here in intervillar space is the maternal blood and the fetal blood is here. And as the placenta matures, these blood vessels will be touching the syncytial trophoblast to decrease the placental barrier. If you talk about villi, you have stem villi and or anchoring villi and free villi or terminal villi. All right, there's some structure called the outer cytotrophoblastic shell. You see that syncytiotrophoblasts, this dark green layer, has the capacity to erode, and it keeps on eroding until someone can stop it. And the only thing that can stop it is this, interestingly, is this layer called the cytotrophoblast. This cytotrophoblast cells therefore have to go through the syncytiotrophoblastic, go outside the syncytiotrophoblastic, and form a shell and stop the burrowing from going further. All right, so now we have, can you see that? Can you see the cytotrophoblast cells have now taken a role outside of the syncytiotrophoblastic and now formed what we now know as this cytotrophoblastic shell. Cytotrophoblastic shell. At this time, the connecting stalk becomes very narrow. You can imagine that the big mesodermal wall has to become as narrow as the umbilical cord. All right. All right. Quickly, the clinical correlates related to this session. If the death of cells in the anterior midline of the germ disc occurs, then there's something called midline craniofacial deficiency, and we call it holoprosencephaly. If there is no sufficient mesoderm that's migrating to the caudal part, we have caudal dysgenesis called cyrinomyelia. And if there's a right-left change because of the genes that are involved in the lefty and the others, then we have situs inversus. Sacrococcygeal teratomas are structures or tumors that are comprising of all three germ layers, and they are because of the remnants of the primitive streak. Now, primitive streak was epiblast alone, but they give rise to all three germ layers. So if primitive streak remains, then you have a primitive streak tumor called the sacrococcygeal teratoma. So summary of week 3, we have gastrulation, formation of notochord, and the early formation of neurulation, or the neural tube formation. The neural tube completely forms when the cranial neuropore and the caudal neuropore close. That happens later. In the trophoblast side, we have tertiary villi and the formation of something called the outer cytotrophoblastic shell. The intraembryonic mesoderm at the end of week 3 differentiates into three, or starts to differentiate into 3 segments called the paraxial mesoderm, intermediate mesoderm, and the lateral plate mesoderm. This is intraembryonic mesoderm. Extraembryonic mesoderm had 2 layers, the parietal layer and the visceral layer. Now the intraembryonic mesoderm has 3 parts, not layers, 3 parts. They're called paraxial part, that is adjacent to the notochord is paraxial. Then you have intermediate mesoderm, and you have lateral plate mesoderm. And that brings to the end of my first session on the embryonic period. Thank you so much. Back to you, Hamida.
Yes, sir. Do we have any questions in the chat box, ma'am? Okay, we don't have. So coming to the end of the session, good evening, everyone. Our honorable speaker, Dr. Stephen Francis, and all the participants. It is such an honor for me to get the opportunity to thank all the dignitaries on behalf of SNO Tamilnadu. I would like to express my gratitude to all esteemed delegates of the webinar for their presence and contribution to make this webinar a great success. I extend my gratitude to our honorable speaker, Dr. Stephen Francis, to take out time from his busy schedule to grace the event and pass on the knowledge to us. Thank you for inspiring and encouraging us with your work, sir. I must thank Dr. Abul Hassan sir and Venkatesh sir for their constant support and the organizing team for working hard to make this webinar successful. Thank you, everyone, and thanks for making the event a great success. Thank you, sir.
Thank you.